Good morning. Today we will continue a previous chapter, Principles of Inheritance and Variations, and topic is Chromosomal Theory of Inheritance. Chromosomal Theory of Inheritance. After Mendel's law, Walter Sutton and Theodore Boveri they studied about the chromosomes behavior of chromosomes. They noticed the several similarities in between two, means Mendelian factors and chromosomes. So they the comparison is that chromosomes occurs in pair as alleles are also occurs in pairs. Homologous chromosomes segregate during meiosis and Mendelian factors segregate at the time of gamete formation. Same thing, different chromosomes orient and separate independently as Mendelian factors. And chromosomes and Mendelian factors both maintain their individuality from one generation to another generation. So this is the chromosomal theory. So on that basis, these factors before uh, mm, the discovery of gene by the Johnson, Mendel's proposed his laws of uh, inheritance, and then he called gene as a factor. And after the discovery of gene, this Mendelian factor is replaced by the Johnson's gene. Uh, Now linkage, and first thing is linkage. Linkage means a group of genes which behaves as a unit. They are known as linked genes. And they collectively transfer from one generation to another generation, and this is known as linkage group. And tendency to two or more genes of the same chromosomes to remain together during the process of inheritance, it is known as linkage. Clear? Linked gene, those are behave like a unit, and the number of genes which are present in linked genes, they are known as linkage group. And the linkage is a process when this linkage group or linked gene are transferred from one generation to another generation as a at a same chromosome. So this is known as linkage. The linkage are two types: complete linkage and incomplete linkage. Complete linkage is exhibited when the genes for a particular character, the genes located on the same chromosomes, do not separate and are inherited together over the generation. It is due to non-break in the gene combination. It is very rare in nature. For example, in Drosophila, the gray body and long wing, they are linked gene, behave like same, and cross with black body and vestigial wing. First generation is formed gray body and long. When it in test cross with recessive parents is known as test cross, as you know. I explained you in previous class. They form same. So this is known as complete linkage because and their gray body vestigial wing and a black body gray body long wing and a black body vestigial wing, the 1:1 ratio. Incomplete linkage example is Mendelian's cross because sometimes in dihybrid cross of when two new combination well formed, so that is known as incomplete linkage, where another character, alleles of another character is not linked with same. For example, in Mendelian crossing, you know that dihybrid cross. Dihybrid cross means two pair of contrasting character. For example, uh, tallness red and dwarf white. Okay, when we cross it in first generation, all of you have known tall and red, and in second generation, ratio is formed 9:3:3:1. Okay, so how it means there is a these two combination is of new combination. Parental combination is tall red and dwarf white. Okay, so this is of tall red and this is of dwarf white. This is recessive, this is dominant. But these two new combination is tall and white, dwarf and red. So it means the tallness and redness, they are independent, they are not linked together. Otherwise, if they will link together, the where is tallness, there should be redness. For example, here gray body with long wing. So this is incomplete linkage. So what are the factors affecting linkage? These are the distance. When strength of linkage is inversely proportional to the distance between linked gene. Factors, distance in strength of linkage is inversely proportional to the distance. Age of organism, with increase the age, chance of crossing over are reduced and hence linkage increases. And X-rays, it reduces the strength of linkage. Increase in temperature decreases the strength of linkage. So all these things are generally reduces the strength of linkage. So what is the significance of linkage? Significance of linkage is it reduces the possibility of variability in gametes. Means variety of gametes are less. Linked and unlinked genes which are present on the same chromosomes are called linked gene, and linkage group, the number of linkage groups in an individual is similar to the haploid number of organism. So that is unlinked genes. Now crossing over. Crossing over means mutual exchange of blocks of homologous genes between a pair of homologous chromosomes is known as crossing over. The mechanism of crossing over, where they cross to each other, that is the mechanism. The types of crossing over: single crossing over, in single crossing over, double crossing over, multiple crossing over. uh, in homologous pair, all of you have known, in meiosis homologous pair is formed when they are, this is the single, this is the double, that is the point of crossing over. Double or triple, that is the double, that is the single, that is the multiple. When these sister chromatids are crossed to each other, so non-sister chromatid, uh, because in same chromosomes two sister chromatids are there, so they are called as sister chromatids. Chromatids of same chromosomes is known as sister chromatid, and chromatids of different chromosomes are non-sister chromatid. When non-sister chromatid are crossed to each other and they exchange their uh, some part of homologous uh, chromatids, so that is known as crossing over, and the point of crossing over is known as chiasmata. Same as factors like linkage, distance, frequency of crossing over is directly proportional to the distance between them. The more distant two genes are in chromosomes, age with advancing age, crossing over frequency generally decreases. X-rays increases the frequency of crossing over. Temperature variations in temperature increase crossing over frequency. So, significance: it provides an inexhaustible store of genetic variability in sexually reproducing organisms. As a result of crossing over, new gene combination is formed because they exchange their part. So, new sequence of the um, base pairs is formed and new sequence makes a new character. So, increase of crossing over, it gives the possibility of variability, and increase of linkage give the possibility of uh, give the reduce the possibility of variability. And next point is sex uh, determination. Sex determination, as you know that in all organism, generally in animal, one is male and one is female. So how they determine the sex, which would be male and which would be female? What is the combination of chromosomes in all of you have all of you known as that chromosomes appears in pairs. So, what combination of chromosomes pairing detect the sex chromosomes? Detect the sex, type of sex, male or female. So, now basis of sex determination, chromosomal determination. So, here is two types of that is when female is homogenetic and male is heterogametic. Female mm, female homogametic and male is heterogametic. No, what is the problem? Homogametic means when both chromosomes are same. homogametic and male heterogametic. ठीक है, तो that is the A type. In this, first is X, X and XY type. Here is XX is female and XY is male. It means both chromosomes are same shape and size in female. So this combination gives female, and when in a pair both chromosomes are different in shape and size, so that is known as male. For example, how? So in this case, female produces one kind of gametes only by these chromosomes. So they are known as XX, and in male produces two kinds of gametes, one with X chromosomes and another with Y chromosomes. So it is male. So female is homogametic. So we can write this AA plus XX. AA means autosomes, and for male that is XY. So this type of sex determination found in human and Drosophila both. Drosophila means fruit fly and human beings both. Another type is second, that is XX and XO type. XX, when both chromosomes form same gamete, the it is in female, and when single chromosomes are present in, that is male. So single chromosomes are present, the another is this 0, it indicates 0 means second chromosomes is absent. Okay, so male. So this type of system is found in grasshoppers, especially in insect grasshoppers. Okay. Now B type. B type is when male becomes homogametic and female becomes hetero. That is another type. When male becomes and female becomes heterogametic. So male is homogametic, that is A, Z, W and Z, Z type. So ZWZ type is just avoid the confusion. If we will use same symbol where female homogametic and male heterogametic, तो there is XX for female and XY for male, और यहाँ कर देते कि XY is female, तो confusion बढ़ जाता. To avoid the confusion, dissimilar alphabets is used for this combination. So in birds male are homogametic. So this is known as male because both chromosomes produce same gamete, and female are heterogametic, means both chromosomes produce two types of eggs. So its combination is A plus Z and its combination is A plus W. और जहाँ पर मेल था ऐसा, तो इट मीन्स कि दे प्रोड्यूसेस टू टाइप्स ऑफ स्पर्म, ए एंड वाई। तो ये इन फर्स्ट टाइप ऑफ सेक्स डिटर्मिनेशन था. नाउ another is another type is ZW, Z0 and ZZ type. Same as female is has single, that is found in some moth and butterflies. So ये this is for female and this is for male. So that is the sex determination. And sex determination in honeybee, it is a special determination. It is known as haplodeploid mechanism. Haplodeploid mechanism means in honeybees the female are queens and workers. ठीक है, they develop from fertilized eggs and having diploid number of chromosomes. And the males, which are also known as drones, so they are haploid and having only one set of chromosomes. So they develop from unfertilized haploid eggs by parthenogenesis. तो यहाँ पर दो तरह का हो गया कि नंबर ऑफ सेट्स ऑफ क्रोमोसोम्स and not by X and Y chromosomes. So the genome in honeybee contains 16 chromosomes for haploid, जो कि drone है in honeybee. Females are two types, queens and workers. Both are diploid. Why? Because they develop from fertilized egg. And drones, which are males, ठीक है, they develop from unfertilized. Unfertilized means they have only one set of chromosomes, n chromosomes. So they have 16 and here is 32 chromosomes, ठीक है. And they develop from parthenogenesis. So they develop into male honeybees with only one genome. In honeybee, one genome develops male and diploid set develops female. Clear? Now some disorders which is related with the that is color blindness in sex linked recessive disorder. Some person has no discrimination of red and blue colors, and this is color blindness. So due to defect in either red or green cone of eyes, the failure to discriminate red and green color. So this defect is due to mutation in certain genes. So 8% males are suffering from this. Sickle cell anemia. Second is sickle cell anemia. It is an autosomal linked recessive trait where there is a due to substitution of 6 codon of beta globin hemoglobin में beta globin chain में in beta globin chain at the sixth place 6 codon is changed GAG to GUG. Clear? So this is the mutant hemoglobin which change in the shape of RBC. The RBC shape which should be biconcave, it changed into a sickle shape. So in this shape, it failed to hold the oxygen also. So this is due to mutation. Phenylketonuria is inborn metabolism. It is a inborn metabolism. Phenylketonuria. अच्छा, phenylketonuria, thalassemia, abnormalities, mutations. We will start a new class because